ये सिलेबस है. So the molecules and their interaction relevant to biology, structure of atoms, molecules and chemical bonds, the composition, structure and function of biomolecules, that is carbohydrates, lipids, proteins, nucleic acids and vitamins. Next is stabilizing interaction, van der Waals, electrostatic, hydrogen bonding, hydrophobic interactions, etc. Next is the principles of biophysical chemistry, pH, buffer, reaction kinetics, thermodynamics, the colligative properties. Next is the energetics, the glycolysis, oxidative phosphorylation, coupled reaction, group transfer, biological energy transducers. The principles of catalysis, enzymes and enzyme kinetics, enzyme regulation, mechanism of enzyme catalysis and isozymes. Next is the conformation of proteins, Ramachandran plot, secondary structure domains, motif and folds. Next is the conformation of nucleic acids, helix A, B, Z, tRNA, microRNA, stability of proteins and nucleic acids, metabolism of carbohydrates, lipids, amino acids, nucleotides and vitamins. Next is cellular organization, the membrane structure and function, structure of the model membrane, lipid bilayer, membrane, protein diffusion, osmosis, ion channels, active transport and membrane pumps, mechanism of sorting and regulation of intracellular transport, electrical properties of membranes. Next is the structural organization and function of the intracellular organelles जैसे कि सेल वॉल, न्यूक्लियस, माइटोकॉन्ड्रिया, गोल्जी bodies, lysosomes, endoplasmic reticulum, peroxisomes, plastids, vacuoles, chloroplast, structure and function of cytoskeleton and its role in motility. Next is the organization of genes and chromosomes, operon, unique and repetitive DNA, interrupted genes, gene families, structure of chromatin and the chromosomes, the heterochromatin, euchromatin and transposons. Next is the cell division and cell cycle. Mitosis, meiosis, their regulation, steps in cell cycle, regulation and control of cell cycle. Next is microbial physiology, growth, yield and characteristics, strategies of cell division and stress response. Next is the fundamental process, DNA replication, repair and recombination, unit of replication, enzymes involved, replication origin, replication fork, fidelity of replication, extra chromosomal replicons, DNA damage, repair mechanisms, homologous and site-specific recombinations. Next is RNA synthesis and processing, transcription factors and machinery, formation of initiation complex, transcription activator and repressor, RNA polymerase and capping, elongation and termination, RNA processing, RNA editing, splicing and polyadenylation, structure and function of different types of RNA and RNA transport. Next is protein synthesis and processing, ribosome, formation of initiation complex, initiation factors and their regulation, elongation factors, termination, genetic code, amino acylation of tRNA identity, amino acyl, tRNA synthetase and translational proofreading, translational inhibitors, post-translational modification of proteins. Next is the control of gene expression at transcription and translation level, regulating the expression of phages, viruses, prokaryotic and eukaryotic gene role of the chromatin in gene expression and gene silencing. आगे देख लेते हैं सेल कम्युनिकेशन एंड सेल सिग्नलिंग के बारे में. होस्ट पैरासाइट इंटरैक्शन, द रिकॉग्निशन एंड एंट्री प्रोसेस ऑफ द डिफरेंट पैथोजेंस लाइक बैक्टीरिया, वायरसेस इनटू animals, plant host cell, alternation of the host cell behavior by pathogens, virus induced cell transformation, pathogen induced diseases in animals, plants, cell fusion in both normal and abnormal cells. Next is cell signaling. इसमें है कि hormones and their receptors, cell surface receptor, signaling through the G protein coupled receptors, signal transduction pathways, second messengers, regulation of signaling pathways, bacterial and plant two component systems, light signaling in plants, bacterial chemotaxis and quorum sensing. Next is cellular communication, regulation of hematopoiesis, general principles of the cell communication, cell adhesion, roles of different adhesion molecules, gap junction, extracellular matrix, integrins, neurotransmission, and its regulation. नेक्स्ट इज कैंसर टॉपिक है कि जेनेटिक रीअरेंजमेंट इन द प्रोजेनिटर सेल्स, द ऑन्कोजीन्स, ट्यूमर सप्रेसर जीन्स, cancer and cell cycle, virus induced cancer, metastasis, interaction of cancer cells with the normal cells, apoptosis, therapeutic interventions of the uncontrolled cell growth. Next is the innate and adaptive immune system, cells and molecules involved in innate and adaptive immunity, antigen, antigenicity, immunogenicity, B and T cell epitopes, structure and function of antibody molecules, generation of antibody, diversity, monoclonal antibodies, antibody engineering, antigen antibody interactions, MHC molecules, antigen processing and presentation, activation and differentiation of B and T cells, B and T cell receptors, humoral and cell mediated immune responses, primary and secondary immune modulation, the complement system, toll like receptors, cell mediated effector functions, inflammation, hypersensitivity and autoimmunity, immune response during the bacterial tuberculosis, parasitic malaria and viral HIV infections, congenital and acquired immunodeficiencies, vaccines. नेक्स्ट देख लेते हैं डेवलपमेंटल बायोलॉजी में क्या क्या दिया है. द बेसिक कॉन्सेप्ट्स ऑफ डेवलपमेंट, पोटेंसी, कमिटमेंट, स्पेसिफिकेशन, इंडक्शन, competence, determination, differentiation, morphogenetic gradients, cell fate, cell lineages, stem cells, genomic equivalence, cytoplasmic determinants, imprinting, mutants, and transgenics in analysis of development. Next is the gametogenesis, fertilization, and early development. The production of gametes, cell surface molecules in the sperm egg recognition in animals, embryo sac development and double fertilization in plants, zygote formation, cleavage, blastula formation, embryonic fields, gastrulation and formation of germ layers in animals, embryogenesis, establishment of symmetry in plants, seed formation and germination. Next is morphogenesis and organogenesis in animals, cell aggregation and differentiation in the dictyostelium, access and pattern formation in Drosophila, amphibia and chick, organogenesis, vulva formation in the C. elegans, eye lens induction, limb development and regeneration in vertebrates, differentiation of neurons, post embryonic development, larval formation, metamorphosis, environmental regulation of normal development and sex determination. Next is morphogenesis and organogenesis in plants, organogenesis of shoot and root apical meristem, shoot root development, leaf development, phyllotaxy, transition to flowering, floral meristems, floral development in the Arabidopsis and Antirrhinum. Next is programmed cell death, aging and senescence. So next is system physiology, plant. Photosynthesis, light harvesting complexes, mechanisms of electron transport, photoprotective mechanisms, CO2 fixation, C3, C4 and CAM pathways. Next, respiration and photorespiration, citric acid cycle, plant mitochondrial electron transport and ATP synthesis, alternate oxidase, photorespiratory pathway. Next, nitrogen metabolism, nitrate and ammonium assimilation, amino acid, biosynthesis. Next is plant hormones, biosynthesis, storage, breakdown and transport, physiological effects and mechanisms of action. Next, sensory photobiology, structure and function and mechanism of the action of phytochromes, cryptochromes and phototropins, stomatal movement, photoperiodism and biological clocks. Next is the solute transport, photoassimilate, translocation, uptake, transport and translocation of water, ion, solute, macromolecules from soil, through cells, across membranes, through xylem and phloem, transpiration, mechanisms of loading, unloading of photoassimilates. Next is secondary metabolites, जिसमें आता है कि biosynthesis of terpenes, phenols, nitrogenous compounds and their roles. Next, stress physiology, जिसमें है कि responses of plants to biotic, pathogen and insects, abiotic, water temperature and salt stresses. नेक्स्ट देख लेते हैं सिस्टम फिजियोलॉजी एनिमल, ब्लड एंड सर्कुलेशन, ब्लड कॉर्पसल्स, हीमोपोएसिस एंड फॉर्म्ड एलिमेंट्स, प्लाज्मा फंक्शन, blood volume regulation, blood groups, hemoglobin, immunity, hemostasis. Next is cardiovascular system, comparative anatomy of heart structure, myogenic heart, specialized tissue, ECG, its principle and significance, cardiac cycle, heart as a pump, blood pressure, neural and chemical regulation of all of the above. Next is respiratory system, comparison of the respiration in different species, anatomical consideration, transport of gases, exchange of gases, the waste elimination, neural and chemical regulation of respiration. Next topic is nervous system, neurons, action potential, gross neuroanatomy of the brain, spinal cord, central and peripheral nervous system, neural control of muscle tone and posture, sense organ, vision, hearing and tactile response. आगे देख लेते हैं कि एक्सक्रीटरी सिस्टम, कंपैरेटिव फिजियोलॉजी ऑफ एक्सक्रीशन, किडनी, यूरिन फॉर्मेशन, यूरिन कॉन्सेंट्रेशन, वेस्ट एलिमिनेशन, मिक्चुरेशन, रेगुलेशन ऑफ वाटर बैलेंस, ब्लड वॉल्यूम, ब्लड प्रेशर, इलेक्ट्रोलाइट बैलेंस, एसिड बेस बैलेंस. नेक्स्ट इज थर्मो रेगुलेशन, कंफर्ट जोन, बॉडी टेम्परेचर, फिजिकल, केमिकल, न्यूरल regulation, acclimatization. Next is stress and adaptation. Next, hydrogen digestive system, the digestion, absorption, energy balance and BMR. Next is endocrinology and reproduction, endocrine glands, basic mechanism of the hormone action, hormone and disease, reproductive process, gametogenesis, ovulation, neuroendocrine regulation. आगे देख लेते हैं, दिया है कि इनहेरिटेंस बायोलॉजी, मेंडेलियन प्रिंसिपल, डोमिनेंस, सेग्रीगेशन, इंडिपेंडेंट असॉर्टमेंट, कॉन्सेप्ट ऑफ जीन, एलील, मल्टीपल एलील, सूडो एलील्स, कॉम्प्लीमेंटेशन टेस्ट्स, एक्सटेंशन ऑफ मेंडेलियन प्रिंसिपल जिसमें आता है कोडोमिनेंस, इनकम्प्लीट डोमिनेंस, जीन इंटरैक्शन, प्लियोट्रोपी, जीनोमिक इम्प्रिंटिंग, पेनिट्रेंस, एक्सप्रेसिविटी and phenocopy, linkage and crossing over, sex linkage, sex limited and sex influenced characters. Next is gene mapping methods जिसमें है linkage map, tetrad analysis, mapping with molecular markers, mapping by using somatic cell hybrids, development of the mapping population in plants. Next is extra chromosomal inheritance, inheritance of mitochondrial and chloroplast genes, maternal inheritance भी इसी में आता है. Next is microbial genetics, that is methods of the genetic transfers जैसे कि transformation, conjugation, transduction, sexduction, mapping genes by interrupted mating and fine structures analysis of genes. Next है human genetics, pedigree analysis, Lord score for linkage testing, karyotypes, genetic disorders. Next is the quantitative genetics, the polygenic inheritance, heritability, its measurement, QTL mapping. Next, mutation, types, causes, detection, mutant types, lethal, conditional, biochemical, loss of function, gain of function, germinal versus somatic mutants and insertional mutagenesis. Next is the structural and numerical alteration of chromosome, that is deletion, duplication, inversion and the translocation, ploidy and their genetic implications. Next, recombination, that is homologous, non-homologous recombination including transposition. डाइवर्सिटी ऑफ लाइफ फॉर्म्स. नेक्स्ट यूनिट है कि प्रिंसिपल्स एंड मेथड्स ऑफ टैक्सोनॉमी जैसे कि कॉन्सेप्ट ऑफ द स्पीशीज एंड हायरार्किकल टैक्सा, बायोलॉजिकल नॉमेनक्लेचर, क्लासिकल एंड क्वांटिटेटिव मेथड्स ऑफ टैक्सोनॉमी ऑफ प्लांट्स, एनिमल्स एंड माइक्रो ऑर्गेनिजम्स. नेक्स्ट, लेवल्स ऑफ स्ट्रक्चरल ऑर्गेनाइजेशन, यूनिसेलुलर, colonial and multicellular forms, levels of organization of tissues, organs, systems and comparative anatomy and adaptive radiation and adaptive modification. Next, outline classification of plants, animals and microorganisms, that is important criteria used for classification in each taxon, classification of plants, animals and microorganisms, evolutionary relationship among taxa. Next is the natural history of the Indian subcontinent जिसमें आता है कि major habitat, types of subcontinent, geographic origins and migration of species, the common Indian mammal birds, seasonality and phenology of the subcontinent. Next is organisms of health and agricultural importance जिसमें है common parasites, pathogens of humans and domestic animals and crops. नेक्स्ट, ऑर्गेनिजम्स ऑफ कंजर्वेशन कंसर्न जैसे कि रेयर या इंडेंजर्ड स्पीशीज, कंजर्वेशन स्ट्रैटेजीज एट्सेट्रा. इकोलॉजी की बात कर लेते हैं. सो इट्स वेरी इंपॉर्टेंट यूनिट. इकोलॉजिकल प्रिंसिपल्स, इन्वायरमेंट है कि फिजिकल इन्वायरमेंट, बायोटिक एबायोटिक इंटरैक्शंस. नेक्स्ट हैबिटेट एंड नीश, कॉन्सेप्ट ऑफ हैबिटेट एंड नीश विड्थ ओवरलैप, फंडामेंटल रियलाइज्ड नीश, रिसोर्स पार्टीशनिंग, कैरेक्टर डिस्प्लेसमेंट. नेक्स्ट द पॉपुलेशन इकोलॉजी, द कैरेक्टरिस्टिक्स ऑफ पॉपुलेशन ग्रोथ कर्व्स, पॉपुलेशन रेगुलेशन, लाइफ हिस्ट्री स्ट्रेटजीज, R&K selection, concept of metapopulation, demes, dispersal, interdemic extinctions, age-structured populations. Next is species interactions, types of interactions, interspecific competition, herbivory, carnivory, pollination, symbiosis. Next, community ecology, nature of communities, community structure and attributes, levels of species diversity, its measurement, ages and ecotones. Next, ecological succession, types क्या हैं, mechanisms, changes involved in succession, concepts of climax. Next is ecosystem ecology, ecosystem structure, ecosystem function in energy flow and mineral cycling, CNP, primary production, decomposition, structure and function of some Indian ecosystems, terrestrial, forest, grassland, aquatic, freshwater, marine and eusterine. Next, biogeography, major terrestrial biomes, theory of island biogeography, biogeographical zones of India. Next is applied ecology, environmental pollution, global environmental change, biodiversity status, monitoring, documentation, major driver of the biodiversity change and biodiversity management approaches. Next is conservation biology, principle of conservation, major approaches to management, Indian case studies on conservation strategies जैसे कि प्रोजेक्ट टाइगर, बायोस्फीयर रिजर्व्स etc. थोड़ा हम इवोल्यूशन एंड बिहेवियर को देख लेते हैं कि एमर्जेंस ऑफ एवोल्यूशनरी थॉट्स जैसे कि लैमार्क, Darwin, concepts of variation, adaptation, struggle, fitness and natural selection, Mendelism, spontaneity of mutations, the evolutionary synthesis. Next is origin of cells, unicellular evolution, origin of basic biological molecules, abiotic synthesis of the organic monomers and polymers. Next is the concept of Oparin and Haldane, experiment of Miller in 1953, first cell, evolution of prokaryotes, origin of eukaryotic cell, evolution of unicellular eukaryotes, anaerobic metabolism, photosynthesis and aerobic metabolism. Next is paleontology and evolutionary history, evolutionary time scale, eras, periods, epochs and major events in evolutionary time scale, origin of uni and multicellular organisms, major groups of plants and animals, stages in the primate evolution including Homo. Next, molecular evolution, concepts of neutral evolution, molecular divergence, molecular clocks, molecular tools in phylogeny classification, identification, proteins of nucleotide sequence analysis, origin of new genes and proteins, gene duplication, divergence. Next is the mechanisms, population genetics जैसे कि population, gene pool, gene frequency, Hardy-Weinberg law, concepts, rate of change in the gene frequency through natural selection, migration and random genetic drift, adaptive radiation, isolating mechanisms, speciation, allopatricity and sympatricity, convergent evolution, sexual selection and co-evolution. Next is brain behavior and evolution, approaches and methods in the study of behavior, proximate, ultimate causation, altruism and evolution, group selection, kin selection, reciprocal altruism या फिर neural basis. आगे और भी दिया है कि basis of learning, memory, cognition, sleep and arousal, biological clock, development of behavior, social communication, social dominance, use of space and territoriality, the mating systems, parental investment, reproductive success, parental care, aggressive behavior, habitat selection, optimality in foraging, migration, orientation and navigation, domestication, behavioral changes. Next is the applied biology में है कि microbial fermentation and production of small and macromolecules. Next, application of immunological principles, vaccines, diagnostics and the tissue and cell culture methods for the plants and animals, transgenic animals and plants, molecular approaches to diagnosis and strain identification. Next is genomics and its application to health, agriculture, including gene therapy, bioresource and uses of biodiversity, breeding in plants and animals, including the marker-assisted selection, bioremediation and phytoremediation, and biosensors. 13 यूनिट देख लेते हैं. मेथड्स इन बायोलॉजी, मॉलिक्यूलर बायोलॉजी एंड रिकॉम्बिनेंट डीएनए मेथड्स, आइसोलेशन एंड purification of RNA, DNA, genomic plasmid and proteins, different separation methods. Next is analysis of RNA, DNA and proteins by 1 and 2 dimensional gel electrophoresis, isoelectric focusing gels. Next is the molecular cloning of the DNA or RNA fragments in the bacterial and eukaryotic systems. Next, the expression of recombinant proteins using bacterial, animal and plant vectors, isolation of specific nucleic acid sequence, generation of the genomic and cDNA libraries in plasmid, phage, cosmid, bacterial, artificial chromosome, VAC, yeast artificial chromosome vectors, in vitro mutagenesis, deletion techniques, gene knockout in bacterial and eukaryotic organisms. Next is the protein sequencing methods, detection of post-translational modifications of proteins. Next, the DNA sequencing methods, strategies for genome sequencing. Next, methods for analysis of gene expression at the RNA and protein level, large-scale expressions such as microarray-based techniques. Next is the isolation, separation, analysis of carbohydrates and lipid molecules, RFLP, RAPD and AFLP techniques. नेक्स्ट द हिस्टोकेमिकल एंड इम्यूनो टेक्निक्स, जिसमें आ जाता है कि एंटीबॉडी जनरेशन, डिटेक्शन ऑफ मॉलिक्यूल्स यूजिंग द एलिसा, रिया, वेस्टर्न ब्लॉट, इम्यूनो प्रेसिपिटेशन, द फ्लायोसाइटोमेट्री, इम्यूनोफ्लोरेसेंस माइक्रोस्कोपी, डिटेक्शन ऑफ मॉलिक्यूल्स इन लिविंग सेल्स, इन सीटू लोकलाइजेशन बाय टेक्निक्स जैसे कि फिश एंड जिश. नेक्स्ट इज बायोफिजिकल मेथड्स, जिसमें आता है मॉलिकुलर एनालिसिस यूजिंग यूवी विजिबल फ्लोरेसेंस, सर्कुलर डाइक्रोइज्म, NMR, ESR, स्पेक्ट्रोस्कोपी, मॉलिकुलर स्ट्रक्चर डिटर्मिनेशन using X-ray diffraction, NMR, molecular analysis using light scattering, different types of mass spectrometry and surface plasma resonance methods. Next is the statistical methods, the measure of central tendency and dispersal, probability distributions, binomial Poisson and normal samplings distribution, difference between parametric and non-parametric statistics, confidence interval, errors, levels of significance, regression, correlation, t-test, analysis of variance, x-square test, basic introduction to the mutrovariate statistics, etc. Next is the radio labeling techniques, detection and the measurement of the different types of radioisotopes normally used in the biology, incorporation of the radioisotopes in biological tissues and cells, molecular imaging of radioactive material, safety guidelines. Microscopic techniques में visualization of cells, subcellular components by light microscopy, resolving powers of different microscopes, macroscopy of living cells, scanning and transmission microscopes, different fixation and staining techniques for the EM, freeze edge and freeze fracture methods for EM, and image processing methods in macroscopy. Next is electrophysiological methods, जिसमें है single neuron recording, patch clamp recording, ECG, brain activity recording, Lesion, stimulation of brain, pharmacological testing, PET, MRI, fMRI and CAT. Next is methods in the field biology, methods of the estimating population density of plants and animals, ranging patterns through the direct, indirect, remote observations, sampling methods in the study of the behavior, habitat characterization, ground and remote sensing methods. ये था सिलेबस ऑफ CSIR नेट लाइफ साइंस. इसको आप कहीं अपने एक A4 शीट पे अच्छे से लिख के अपने स्टडी टेबल पे आगे पेस्ट कर सकते हो, क्योंकि कोई भी एग्ज़ाम को स्टार्ट करने से पहले सिलेबस और उसके PYQs हमारे टेबल पे होने ही चाहिए. दैट इज़ मस्ट.